Our second lecture for week 2 is on information transmission, synapses. A synapse is a point of communication between a neuron and another cell. As I mentioned in an earlier lecture, that cell could be muscle, it could be another neuron, or it could be gland. There are different places on a neuron where synapses take place. Axodendritic synapses are the most common. This is where one neuron synapses on the dendrite of another neuron. Axosomatic is faster and is probably more common in our sensory pathways than in other pathways, but it's not as common as axodendritic. Axosomatic is where a neuron synapses on the soma, or cell body, of the next neuron. And then axoaxonic would be the fastest synapse as far as having an effect. And this is involved when you need lots of recruitment. You've got to contract a muscle really, really, really strong, so you want lots of neurons to that muscle firing. So your central nervous system is going to recruit a lot of neurons to go to your muscle. And so here we might see more axoaxonic synapses going on. This is very common with interneurons. Most of the time, an axoaxonic synapse is going to take place right near the axon terminals in the telodendria. The axon terminal is also known as a presynaptic terminal. At the level of the terminal, there are voltage-gated calcium channels in the membrane. There are many of these. Within the axon terminal, we have synaptic vesicles. Synaptic vesicles contain neurotransmitter. These synaptic vesicles are held in place in the axon terminal by something called synapsin. The synaptic cleft is a very small space between the axon terminal and the receptor on the postsynaptic cell. So your neuron is your presynaptic neuron. Then you're going to have a little gap, the synaptic cleft, and then you're going to have another cell, and that would be your postsynaptic cell. If it's a neuron, it would be a postsynaptic neuron. And as I mentioned, that postsynaptic cell has receptors on the membrane, and receptors are really specialized proteins. Your action potential that we talked about in the last lecture has now propagated down to reach the axon terminal. When it reaches the axon terminal, it causes the opening of the voltage-gated calcium channels. This results in an influx of calcium into the axon terminal. The calcium then triggers a sequence of a bunch of chemical reactions that we're not going to get into, and this causes synapsin to release the synaptic vesicles. The synaptic vesicles then bind to the membrane of the axon terminal. These vesicles are actually made of the same stuff. They have that same lipid bilayer construction as the membrane. So when they float down to and come in contact with the membrane, they simply bind to it. This allows them to expel their contents, expel the neurotransmitter into the synaptic cleft. And that process of expelling their neurotransmitter into the synaptic cleft is called exocytosis. The neurotransmitter then diffuses across the synaptic cleft and binds to a postsynaptic cell receptor. There is a small delay for the neurotransmitter to get across the synaptic cleft. In other words, it's not an immediate, as soon as it's released, it's binding. It has to diffuse through the fluid across that synaptic cleft. And this is called the synaptic delay. We really cannot change that synaptic delay, so we can't try and speed up information transmission by decreasing that delay. And there's a video here on action potentials and synapses that might help with your understanding of what's going on at the synapse level. Here is a drawing to help us understand the synapse. So we have here an axon terminal in yellow. We have the postsynaptic cell in green. In pink on the postsynaptic cell are special receptors. In brown, in the presynaptic cell on the axon terminal, we have voltage-gated calcium channels. Those large white circles with the little circles inside would be synaptic vesicles filled with neurotransmitter. And then there's some other things on here that we really don't need to get into too much. But what you have is your action potential comes down. It causes the opening of the voltage-gated calcium channel, which allows calcium into the axon terminal, which causes the synaptic vesicle to be released by synapsin and move to the membrane of the axon terminal, where it binds to it, opens up, and releases the neurotransmitter into the synaptic cleft. The neurotransmitter then binds to the receptor on the postsynaptic cell. And when it binds, that's going to trigger the opening of ligand-gated channels. Depending on what type of ligand-gated channel opens will determine if we get a depolarization response or a hyperpolarization response. So our action potential is an electrical event, the movement of ions across the neuron membrane. The synaptic cleft sequence is a chemical event. We have a chemical neurotransmitter diffusing across the cell to bind to the receptor. When the neurotransmitter binds to the receptor, if we have a small depolarization, remember these are local potentials, so they're not big. If we have a small depolarization, that is going to be excitatory. If we have hyperpolarization, that is inhibitory. That local potential that takes place is called a postsynaptic potential because it's taking place after the synapse. An excitatory postsynaptic potential is known as an EPSP. An inhibitory postsynaptic potential is known as an IPSP. When you have an EPSP, an excitatory postsynaptic potential, the binding of the neurotransmitter is going to cause the opening of a ligand-gated channel that's going to cause an excitatory or depolarization change. So the postsynaptic cell is going to become less negative. Examples would be to open up a ligand-gated sodium channel or a ligand-gated calcium channel, causing the particular ion to influx into the cell. Now, average EPSPs are quite small. They're only 0.2 to 0.4 millivolts or so. So we generally need about 50 to 100 of these in order to reach threshold, assuming that at the same time we're not getting any inhibitory synaptic potentials. That's one reason we have lots of receptors going on a particular cell, so that we're getting lots of these local potentials we can add up spatially. And we're also able to pull off a neurotransmitter molecule and then have another one take its place. So temporally, we can also summate. So we summate up. We get 50 to 100 of these guys to summate up in order to reach threshold. If we don't reach that threshold, we will not generate an action potential in the next cell. So here's our picture. We have our minus 70 millivolt resting membrane potential. We have minus 55 millivolt threshold. We have the stimulus, so the neurotransmitter binding. One neurotransmitter binding to one receptor creates an excitatory postsynaptic potential of about 0.2 to 0.4 millivolts, so I put it right in the middle at 0.3. So now we're down to minus 69.7 millivolts. Obviously, we're not anywhere close to threshold, but if I can add up a bunch of these, I will hopefully achieve threshold. An IPSP is going to do the opposite. It's going to cause the postsynaptic cell to become more negative inside, to be hyperpolarized. This occurs if we get the opening up of potassium channels or ligand-gated chloride channels. So potassium leaves the cell or chloride enters the cell. Either of those things creates hyperpolarization, making it more difficult to reach threshold and generate an action potential in this postsynaptic cell. And you can see that here. We have our stimulus, and instead of now being less negative, we're 0.3 millivolts more negative than where we started. We're getting further away from threshold. Now, we have approximately 100 billion neurons in our nervous system. Each of these neurons has anywhere from 1,000 to 10,000 synapses. And in fact, I recently read that we have some neurons in our cerebellum that have as many as 150,000 synapses on a single neuron. It's just amazing. This is a phenomenal number of possible combinations. So what's going to happen? How do we know if we're going to get an action potential or not in that postsynaptic cell? Well, it's all the balance. It's what's going on at that moment in time. It's the balance of our EPSPs to our IPSPs. And that, again, is changing from moment to moment. If we have more active EPSPs and we have enough of them that we get a net summation to reach minus 55, then an action potential is generated. If there are some, though, of our positives and negatives, if our EPSPs plus our IPSPs ends up being less than 15 millivolts, or we started off at a point where we needed to have a bigger change than that, and we don't reach threshold, no action potential will be generated. The goal of some neurons is to minimize or limit the number of action potentials generated by the next cell. So these will release neurotransmitters that are going to bind to channels to create IPSPs because they don't want us to have as many action potentials in the next cell. Others want the opposite effect. So it's the balance of what's more active at the time. Synaptic receptors can be divided into direct-gated and indirect-gated receptors. Direct-gated receptors are basically what we've been talking about. You have a protein that acts as the receptor and binds to the neurotransmitter in the synaptic cleft. It simultaneously acts as the ion channel. So when the neurotransmitter binds to it, it causes the protein to change shape. This is called a conformational change. When the protein changes shape, that opens up the channel, allowing the ions to move through. When you remove the neurotransmitter from the receptor, the protein goes back to its original shape, which closes the gate, and the ions cannot move through. This is a very fast process. As soon as the neurotransmitter binds, the gate opens, ions move through. Here you can see the example. My receptor is on top of, I have receptors on top of the gates. You can see the particular gate on the right. The neurotransmitter in the red triangle has bound to the neurotransmitter there, which immediately caused the gate to open. And ions can now move through. Indirect gated receptors, on the other hand, are going to work more slowly. Here we have a receptor in the synaptic cleft to bind to the neurotransmitter. However, this protein, while it acts as a receptor, does not act as a gate. It doesn't change shape to open up a space to allow ions to move through. As a result, there's going to be a multi-step process for us to open up our gates, and this process then is going to be slower than with direct gated mechanisms. What we have with this protein, there are different types, but I'm going to talk about the G protein. So if our indirect gated receptor is a G protein, inside of the cell, the protein has different subunits. Of these subunits, one is called an alpha subunit. Basically, it's alpha, gamma, beta, I believe. The binding of the neurotransmitter causes a conformational change in the G protein, but instead of that opening up a channel, it causes the release of subunits on the inside of the next cell so that they start diffusing through the intracellular fluid. As they move through the cell's cytoplasm, multiple things happen. You get a cascade of events that triggers changes in things like RNA expression and cellular functioning, but you also get the alpha subunit binding to a receptor on the inside of the cell membrane, and this particular receptor does act as a gate. This causes the second receptor then, that protein, to change shape, opening up the gate to allow the movement of ions through. All right. So again, we get a bunch of other things going on, such as biochemical reactions with cyclic AMP, turning on of genes, and other kinds of things. And this will become a little bit more important later when we talk about learning next week, because you need to have this turning on of genes in order to create new and more synapses. And that is required for long-term learning. But here's a picture of our indirect gated mechanism. So we have a G protein in the membrane. It binds to the neurotransmitter in the synaptic cleft. On the inside of the neuron, we have some subunits on this G protein. Those subunits, the only one I want you to know the name of is the alpha subunit, but they are released from this G protein when the neurotransmitter binds to it. The alpha subunit is going to move through the cytoplasm and bind to a receptor further down the membrane. That receptor acts as both the receptor and the gate, changes shape, opens up the gate, allowing ions to pass through. It is the postsynaptic receptor that determines whether a given neurotransmitter is going to have an excitatory effect or an inhibitory effect. The same neurotransmitter in some cells will be excitatory and in others will be inhibitory, because it all depends on the type of receptor it binds to. This is a nice quote that says it all in a nutshell. A single chemical messenger can evoke either an EPSP or an IPSP, depending on the receptor to which it binds. Different neurotransmitters are categorized by their chemical structure as well as their size. We have small molecule neurotransmitters and large molecule neurotransmitters. Small molecule neurotransmitters are not proteins. They can be produced pretty much anywhere in the neuron, either the cell body or in the axon terminal. They're most likely going to be produced in the axon terminal. They require less calcium into the axon terminal for exocytosis to take place. Our large molecule neurotransmitters, on the other hand, are peptides and proteins. They need that mechanisms involving the messenger RNA and then transcription RNA and Golgi bodies and rough endoplasmic reticulum. They need all of those organelles in the soma of the cell in order for manufacture. So these guys have to be produced in the soma and then shipped down to the axon terminal. They require a higher amount of calcium for exocytosis. There are several neurotransmitters I want you to become very familiar with, because we'll be talking about them extensively throughout the semester. The first is acetylcholine, abbreviated ACH. Acetylcholine is found at the neuromuscular junction, and it is always excitatory at the neuromuscular junction. It is also found, however, in many other places of the central nervous system. Most of the time, you will find that the receptors result in excitatory response. So acetylcholine, think excitation. That's not 100% true, but it is most of the time. Receptors for acetylcholine are called cholinergic receptors. Dopamine is found in many places of the brain as well. Dopamine is known as the pleasure juice because it is released either in anticipation of or upon receiving a reward. It helps us to feel really good, which is one of the reasons why it ends up being involved with addictions, because with most addictions, you get a large release of dopamine, which gives you a high, and then you want to repeat that high because it feels so good. But dopamine is not just related to reward. Dopamine is involved with movement and cognition, so it's quite an important neurotransmitter and found in many places. It has both excitatory and inhibitory receptors, so it's very dependent upon the type of receptor it binds to as to whether it will generate IPSPs or EPSPs. Epinephrine and norepinephrine are very important in our sympathetic nervous system, as well as being involved in other parts of the nervous system. Norepinephrine in particular is involved with many neurons in the brain and brainstem. These can be either excitatory or inhibitory, depending upon their receptor. Serotonin is often involved in regulation of mood and pain, and again, its receptors can either be excitatory or inhibitory. Glutamate is probably our most important and most profound, I guess it's not quite the word I'm looking for, but maybe extensive, excitatory neurotransmitter when it comes to the central nervous system. Acetylcholine obviously is very important in the peripheral nervous system at the neuromuscular junction. Acetylcholine is often also a major neurotransmitter in our autonomic nervous system. But in the central nervous system, glutamate is a very important excitatory neurotransmitter, and glutamate is critical for long-term learning. It has two types of receptors that you will be reading about. One is called the NMDA receptor, and the other is an AMPA receptor. GABA, on the other hand, is probably our most important inhibitory neurotransmitter. So when you think glutamate, I want you to think excitatory. When you think GABA, I want you to think inhibitory. As you saw, many of the others could be either, depending on the receptor. Then we have endogenous opioids, such as beta endorphins, enkephalins, and dynorphins. These have predominantly inhibitory responses when binding to their receptors. Opioids are very important in the perception and modulation of pain. Morphine is a type of opioid. Substance P is found in the spinal cord and brain and is involved in the perception of pain and inflammation. It has predominantly excitatory responses. And nitric oxide, which is a very small molecule neurotransmitter that results in smooth muscle relaxation and vasodilation, and is involved with learning and memory. It is produced, again, mostly in the axon terminals, being a small molecule. Your book is going to talk about the difference between a neurotransmitter and a neuromodulator. So what's the difference? Well, a neurotransmitter is released directly into the synaptic cleft. It has an effect at a single synapse. It doesn't go around and go through the extracellular fluid and create effects at all kinds of neurons. It has an effect at a single synapse. And that effect is going to be relatively short term. We're going to take these neurotransmitters, we're going to break them down, or we're going to remove them, and the effect is over. A neuromodulator, on the other hand, is released into the extracellular fluid surrounding the axon terminal, but away from the synaptic cleft. It diffuses through the extracellular fluid and goes to several neurons, sometimes at a distance away. So it's acting on several neurons instead of just a single synapse on one neuron. As a result of needing to diffuse to get to these other neurons, it takes longer before it becomes effective. But that effect also tends to last much longer, lasting from minutes up to even days. So how do we take neurotransmitters and get rid of them, removing them from the receptor in the synaptic cleft and getting them out of that synaptic cleft when we're done transmitting our information at that particular synapse. Well, there are three primary methods. There is diffusion out of the synaptic cleft, and when that happens, you're going to see that astrocytes, those glial cells we discussed last week, are going to come around and take up this excess neurotransmitter that's diffused out of the cleft. Enzyme degradation, for instance, with acetylcholine, you have acetylcholine esterase. That's an enzyme that breaks down acetylcholine. So it literally degrades the neurotransmitter. Or you can have reuptake, which is probably the most common removal method. With reuptake, the neurotransmitter is pulled off of the receptor and then pumped back into the presynaptic terminal. There it's either broken down and good parts are reused. It may be transported back up to the soma, so the soma can put it back together with all working parts, or it can just simply be broken down and gotten rid of. We can change the number of receptors that are located in the postsynaptic cell, and this is going to change depending on how much the synapse is used and if it's used too much, too little, or the intensity of its use. Downregulation is when we have fewer active receptors on the postsynaptic cell. Either we inactivate receptors, so we attach something to the receptor to make it so it doesn't work, or we literally can take the receptor, pull it out of the cell membrane, and either degrade it or store it, so that if we want it later, we can pop it back into the membrane. Downregulation often occurs in the case of overstimulation. For instance, the first time someone takes a drug, there is a huge release of dopamine. This release of dopamine is so large that the postsynaptic neurons actually take receptors out of their membrane and downregulate. As a result, the next time the person takes that drug, there are fewer receptors, and the response in the next cell is lessened because it is a protective response. If we were to have this overstimulation all the time and these huge results, we would end up being toxic to the neurons. So as a protective response, the next neuron downregulates receptors. They say that the first high is the best high. And after that, when someone is addicted, they're always trying to get back to that first high, but they can't because of this downregulation. Downregulation, as I said, leads to less sensitivity in the next cell, which is also called tolerance. We can also upregulate. We can put more receptors into a cell membrane, or we can activate cell receptors that had previously been inactive. This is often a response to understimulation. Ooh, there's not enough neurotransmitter here. I better put in more receptors so that I can grab every possible molecule of neurotransmitter because I'm just not getting enough of them. This results in an increased sensitivity. We'll also see this not just with understimulation, but with repetitive use. So the more you use that synapse in a normal way, not overstimulating, but you use it in a normal way, and there's times when it's not used and other times when it is, so it's not a constant. Constant stimulation will tend to lead to tolerance and downregulation. But if you have a normal use, but you use it a lot, you will see an upregulation and an increase in sensitivity. Now, what about drugs on synapses? Well, drugs can be either agonists or antagonists. An agonist mimics the role of a neurotransmitter. So if I give you morphine, that is going to bind to an opioid receptor, mimicking the response of if your body had released beta endorphin on its own. So an agonist mimics the effect of the neurotransmitter. An antagonist blocks the neurotransmitter. It either binds to the receptor and doesn't have any effect, but that because it's bound to the receptor, the neurotransmitter cannot bind, or it prevents the neurotransmitter from being released from the presynaptic neuron. We also have what are called reuptake inhibitors. In order to take a neurotransmitter and reuptake it into the axon terminal in order to reuse it, we have to have a pump. It takes energy and an active pump that pulls that neurotransmitter off of the receptor and puts it back into the presynaptic axon terminal. We can block those reuptake pumps, and this is called a reuptake inhibitor. Probably one of the more common ones is for serotonin. Individuals with depression may be given a serotonin-specific reuptake inhibitor, commonly known as an SSRI. So a serotonin-specific reuptake inhibitor blocks this reuptake pump. Serotonin stays in the synaptic cleft longer because it can't get back up into the axon terminal as easily. And so it can have greater effect on the next neuron, and that helps with mood. So now some brainstorming. What would you expect to happen if you had too little acetylcholine at the neuromuscular junction? What would you expect to happen if you had too many or too few acetylcholine receptors on the sarcolemma? So if you might remember from biology, the sarcolemma is the muscle cell membrane. So in the first case, the problem is the neurotransmitter. There's not enough of it being released. In the second case, it's a problem with the number of receptors on the postsynaptic cell, in this case, the muscle cell. Let's put this on this week's discussion question and answer board. And now a pathology. I'm only going to talk about one. That is myasthenia gravis. Myasthenia gravis is a destruction of acetylcholine receptors on muscle cells. So we don't have enough receptors. Or it might be that the receptors are somehow damaged so they can't bind appropriately to acetylcholine. Myasthenia gravis is found in both men and women. It is usually diagnosed much earlier in women, however, generally in their second decade of life, whereas men, it's usually not until about their sixth decade of life. With myasthenia gravis, an individual will experience progressive weakness with repetition of an activity. They need to have frequent rest breaks, so they'll improve after rest. But once they start up again and they continue to do an activity, they will get progressively weaker and weaker. So that's a very kind of classic muscular response with when you have an individual with myasthenia gravis. And something you need to keep in mind when planning your treatment interventions is that they will need very frequent rest breaks. And you don't want to do too many repetitions at one time. So that's the end of the second lecture and the last lecture for this second week of our Unit 1. Next week, we're going to be talking about neuroplasticity and changes that take place in the nervous system with aging.